Hello friends, today we are going to study DNA cloning. Okay, so what is DNA cloning? As the name suggests, DNA, as you people know what is DNA. Now, what is cloning? Cloning means to make copy. Okay, to make copy of something. To make clone. Clone means exact copy. Okay, so here what we are going to do is make copy of DNA, right? Okay, now how we are going to do this? What are the basic things that we need? Okay, first we need a vector. Vector. Now what is vector? Vector is an independently replicating DNA sequence. Okay, means it doesn't need anything for its replication from others. Okay, it doesn't need anything from others for itself. It can replicate by its own. It has everything, every type of protein, enzyme that it needs to replicate itself. It has everything. Okay, it is circular. Okay, now examples, there are various vectors. There are bacterial vectors, then there are plants vector, animal vector, etc, etc. We'll learn detail on vectors in future episodes. Okay, for now just remember this, that vector is a DNA sequence that can replicate independently. Okay, now second thing that we need is gene of interest. Okay, now what is gene of interest? Means the gene that we have to amplify, that is known as gene of interest. Suppose this is a DNA sequence and this is a gene, very important gene it is, and we have to amplify this gene to get more and more product of that particular gene. Suppose this gene encodes insulin, as insulin is very important nowadays, so for diabetes patients, so we have to amplify this gene and we have to get more and more product of insulin. uh, we have to get more and more product of this gene, that is insulin. Okay, so we will amplify this gene. Okay, insulin is just a example. The gene could be of anything. It could be of growth hormone, it could be of any other protein, anything. Okay, so now what we have to do is we have to insert this gene into this vector. Okay, so suppose this is a vector and we will insert this particular gene into this vector. Okay, now how we are going to do this? We'll use one restriction enzyme for both, for vector as well as for gene of interest. We'll cut this restriction. We'll use one restriction enzyme. Suppose it is equivalent. There are various type of restriction enzyme. And each restriction enzyme has one particular restriction site. So it's very important that that restriction site should be present in vector as well as in your gene of interest, because we are going to cut both of them with the same restriction enzyme. Okay, so this restriction enzyme we use to cut this vector. Okay, so it make this it will make this type of cut. Okay, then it will open up this circular DNA. Okay, then we will use the same restriction enzyme for to cut our gene of interest. Now the question arises is why we are using the same restriction enzyme? Because this restriction enzyme will make the ends of our gene, this gene and the end of this circular DNA would be complementary to each other and they will fix into each other. The detail of how restriction enzyme works, what are the restriction enzyme that we use in DNA cloning method, we'll learn about this later. For now just remember that the same restriction enzyme is used to cut the vector as well as to gene of interest. Okay, now when we then we will insert this gene of interest into this restriction enzyme. Okay, now the product that we will get is known as recombinant DNA. Okay, recombinant DNA. Okay, so then we will means the we'll get this recombinant DNA. Now this recombinant DNA will insert into the host cell. The host could be E. coli, it could be yeast, and etc. Okay, so why we are using E. coli or yeast or this? Because the duplication time of E. coli is very short, that is of 20 minutes. That is within 20 minutes the E. coli will get duplicate, and as E. coli will get duplicate, our gene of interest will also get duplicate. Okay, so suppose we have inserted this this our gene of interest here. Now inserted our recombinant DNA into this host. Okay, now within 20 minutes this E. coli will get duplicate, right? As E. coli will get duplicate, our gene of interest will also get duplicate. This is within 20 minutes. Okay, then later in next 20 minutes again we'll get this E. coli duplication copies of E. coli. Okay, now as this E. coli has short replication time, it will get duplicate easily, and as E. coli get duplicate, our gene of interest will also get duplicate. So therefore we are using this E. coli or yeast as our host. Okay, so in this way it's the basic overview of DNA cloning. The detail of what are the vectors we are going to use for different types of gene, what are the enzymes that we are going to use to insert our gene of interest into the vector, etc. We'll learn about this later. Okay, in the next future lectures. So if there is any doubt, if you have any doubt, please comment. Thank you so much.